Our next panel focuses on a unique model and partnership with UCLA. I am so pleased to welcome Dave Licata, president of Culver City-based TORL Biotherapeutics, along with new company CEO, Aran Maree. Also joining them is Dr. Dennis Slamon, Director of Clinical Translational Research and Director of the Revlon UCLA Women's Cancer Research Program at UCLA Health Johnson Comprehensive Cancer Center. Please join them and welcome them to the stage.
So we'll get started here. I wanted to take just a few minutes at the beginning of the presentation to thank some folks. The TORL was founded in 2019, and we're going to take you through that story. But there are a lot of people in this room and a lot of people at UCLA and at the Regents who were part of the TORL story. So thanks for allowing us to present our story today. We'd also like to thank UCLA and TDG for supporting this from the very beginning. Amir worked with us to build a new model for university translation we're going to talk about today. Thanks also to Mark and to Dan Oscar for everything that TDG does to advance this. We'd also like to thank UCLA leadership for again, believing in us and supporting this new model that we're here to talk about. In particular, Judy Gasson, John Mazziota, Jonice Spiso, Steve Dubinet, and Julio Frank. It has been a real pleasure over the last 6 years to watch things move and for this model to advance. And it really did involve help from everybody. Finally, I'd like to thank Jagdeep Bakar, Larry Atkinson, and Steve Sternum from UC Investment in Oakland, who've been by our side, have been really committed to watching these drugs move forward into the clinic and providing support for that to happen. So starting a little in reverse with a thanking, but I think you'll understand why as we go through this story. It is an unusual model. So we were founded in 2019, and we've raised $455 million to date. As everybody in this room knows, clinical trials are expensive. They're particularly expensive when you are a multi-asset company from day one. I'd like to point out and congratulate Aran. It's been a big month for us. Aran is our new CEO. He's also wearing the CMO title as well, at least for a time being. But thank you, Aran. Mark Alez remains our chairman, and we're looking forward to the next couple of years. But congratulations, Aran. So when we started the company, when Dennis and I founded the company with our co-founders in 2019, the mission was pretty simple. It was to deliver multiple drugs to patients as quickly as possible. So the challenge with that is for a startup company doing drug development, you generally focus on a single asset. We didn't want to do that. Right off the bat, we wanted to be working on multiple, and we wanted to imagine a world where it was multiple every year. The idea was to build the overlap that Chancellor Frank discussed this morning, right from the beginning, where the Slamon lab and TORL Biotherapeutics were intertwined, and there was an ongoing and enduring relationship between the lab. That's atypical for a startup. Usually what happens is that the research that the startup is launched from then goes its own way, generally fairly separate from the university. But the idea here was, in order to be multi-asset, in order to really have access to the world-class science coming out of Dennis's lab, is that we would lean into the university model. And what that really meant is that we would lean into discovery happening in the Slamon lab, and that TORL would then translate it. Another way of saying that is what we wanted to do is we wanted to build a scalable value creation engine. And so for value creation, we were defining value creation as value creation to patients, first and foremost, value creation for the university, where all this research is coming from, value creation to our investors who are betting on us and supporting us. That was particularly important, particularly in the early years, because the pitch that Dennis and I would make to our early investors is, we want to put 5 drugs in the clinic in the next 3 years, but we only need $20 million to start. So incrementally we proved ourselves, and the investors believed in us more and more, and to date, $455 million later, we are still doing what we were trying to do from the beginning. And finally, value creation for our team members, for the employees at TORL who are working so hard to get this done. The name TORL And this really speaks to how intertwined and interreliant we wanted to be with the Slamon lab. Is Dennis's lab is called the Translational Oncology Research Lab. TORL is TORL Biotherapeutics. In order to do this, we worked with Amir Nyberg, who you've heard from today, to rethink how license agreements work. And in our case, what that meant is that we would build a license agreement that would work not only for the first two assets that we had when we started in 2019, but would be the mechanism for additional assets to come into the company over time. The way we describe this is it's frictionless licensing. When we got to the point where both TORL and Dennis and the lab decided it was time to move into the company, it was a simple email from both us and the lab that let TDG know we're moving forward with another program. We've done that 6 times so far. It also was a commitment to significant ongoing sponsored research. So we do about $2 to $3 million a year in sponsored research at UCLA and Dennis's lab. And really the best way to think about that is we don't have our own basic lab at TORL. The discovery happens at UCLA, and we're committed to that as our model, and so we haven't built out our own lab. And so as such, a big part of our budget is allocated to the sponsored research program. It's worked very well. We've been doing it for 6 years, and we've refreshed the agreement, so we're going to be doing it for another 4. It is a big part of the model, an important part of the model. When we thought about being a multi-asset company, we also thought about the company structure. This is a little bit in the weeds, but I do think it's important. As different than almost every startup in therapeutics or beyond therapeutics who are C Corps, we're an LLC. The reason why we're an LLC is we wanted the option of being able to exit a program without exiting the whole company. That's difficult to do. A lot of the biotech acquisitions that you see are acquisitions for their lead program, but the entire company is acquired. It's generally cheaper to acquire the whole company than a single asset in the way that tax and structure works. So we were hoping, if we wanted to ever exit a program, that we would get to keep working on our follow-on programs. In other words, our early programs are as important to us as our late programs, and we want to invest across all of it. We also wanted to keep the team together. What we were doing with the Slamon lab, what we were doing inside the company, everybody working together, we wanted to be able to keep doing it the next day. We don't know whether we'll ever use this option, but we wanted the option, and that's why we structured ourselves as an LLC. Everybody talks about being patient-centric, and I think everybody hopefully in this room is. For us, patient-centric means something very specifically, which is that of the $455 million we've raised, we try to make sure at least 80% of that is going to either manufacturing drugs for patients for clinical trials or for paying for the clinical trials. It is trying to keep our overhead low, being capital efficient, and what that allows us to do is to think as big as possible on each one of our assets. So you'll hear about XOV today. We not only are focused on ovarian cancer, but also non-small cell and testicular cancer. And we're not only focused on where we think our first indication will be, which is in the later lines, but we're actually dosing patients now who are newly diagnosed with ovarian cancer with our drug alongside standard of care. Again, these are hard things to do for a smaller company, but we feel like they're important, and that's how we define being patient-centric. So another way of saying this is we try to align interests for common success. If we win, UCLA wins. If UCLA wins and the Slamon lab wins, the company wins, and patients win. And so we really are benefiting from world-class research. We're benefiting from potentially world-class drugs. And UCLA is with us along the way. Everything that we touch, UCLA is part of. So how does that actually work? Well, actually, the target discovery and building the antibody ADC itself is all done in Dennis's lab. At a certain point, as the lab starts to believe that there's potential there, the company starts applying sponsored research dollars to the program. Generally, even before we license the program, we'll start spending money to better evaluate it or to support research that's generally not done in the lab, that's done outside the lab, in order to best evaluate the program and to prioritize the many programs coming out of the lab. At a certain point, if we reach a threshold where we believe it should go into patients, that's when we use this framework license that TDG, Amir, and all of us developed in 2019 to bring it into the company. At that point, we start manufacturing. That is something we do together. So the same researchers who developed the antibody at UCLA will sit on calls through the sponsored research agreement with ourselves at TORL, but also with our manufacturer. We're scaling from pretty small scale to kilogram scale, and we're trying to do this quickly. The best way of describing how well this has worked is that we can go from deciding to start manufacturing to filing an IND on an ADC, on an antibody drug conjugate, in about 16 months. So it's about a year faster than is typical with our peers. And that's because we try to make sure that every week or two when we're on these calls, we can answer the questions by our manufacturer partner and make sure they can move the ball forward as quickly as possible. In fact, one of our programs was one of the first examples of them applying their fast to antibody manufacturing program they developed for COVID antibodies to oncology antibodies. And they identified us as the group they wanted to start with because of how quickly we move. This is the idea that patients are waiting. And again, once we're in the clinic, TORL is really driving this, the patient's accrual. But at the same time, the lab at UCLA is continuing to investigate the target and the drug preclinically, and one feeds the other. The clinical data feeds what the kind of research being done is through sponsored research in the lab, and that basic science and preclinical research is helping us to decision make with clinical decisions. Ultimately, we hope we're in the position to start a phase 2, a registrational, hopefully trial, and a phase 3 and be commercial, and this is where a lot of money gets spent. And this is where we kind of take things forward. We're in that right now, and we hope to be at the commercial stage in the next few years. Right now, we have 4 active programs. We have had a total of 6 programs. 2 have been deprioritized in 2025. We're going to talk about XOV, our Claudin-6 ADC today. Dennis is going to take you through how we identified the target, prioritized the target, and Aran's going to take us through our clinical experience to date. We also have DLK1. We don't have time to talk about that today, but that is in small cell lung cancer and also sarcomas. We're just finishing with dose escalation and moving into expansion right now. We also have just started clinical work, escalation work on what we're still not disclosing, which is a GPCR target that we have drugged with an ADC. To our knowledge, we're the only one in the world working on this target, and we therefore are keeping it pretty quiet so far. But we're quite excited. Dennis will talk about why he picked that target to work on, and also how difficult it is to drug a GPCR target with a biologic. Finally, as of this month, we're bringing a new program in. So we hope to start manufacturing. When we bring it in, we start quickly. We're going to start manufacturing hopefully in 2 to 3 weeks on this program. This is going to be for pancreatic cancer, for gastric cancer, and also non-small cell lung cancer. So with that, I'm going to turn it over to Dennis to talk about the lab. I would like to just remind everybody of how impressive a lab that it is. It's 25,000 square feet. It is 5,000 animal vivarium. It is almost a biotech inside UCLA. That's part of why this works so well. It is a lab that has generated, as Dean Dubinet said, really amazing advancements for women with breast cancer through Dennis's work with Genentech and with Pfizer over the years. And so very long before TORL was around, this was a very productive lab, but it's been a real pleasure to work with Dennis.
So thanks, Dave. I'll take you briefly through the platform that we use. You could take this story as a Xerox copy for all of the other targets we're taking forward. Dave mentioned we wanted to be a multi-asset company at the outset. It's not based on a single gene or a single pathway, but what the platform was able to tell us about what our potential targets, and that is potential for target identity. Once a target is identified as potential, it has to be validated. So the lab also can do the validation process, and we have the appropriate people in place to be able to do that, all done in an academic discovery lab. And even the generation of the antibodies now are no longer done in terms of the research-grade antibodies, not the commercial-grade antibodies. Those are also done within the laboratory so that we can show that the therapeutic that we're trying to develop and the sequence for that antibody really is efficacious and does have an appropriate safety profile, at least in the preclinical models that we have. People talk about this being the Slamon lab. I want to say at the outset, nothing could be further from the truth. It is a group of investigators led by some lead scientists like Martina McDermott, Neil O'Brien, Ben Hofstrom, and a whole team of career technologists who work within this company, about 22 in total, that make this happen. And we pair up with our own clinicians from UCLA who know the disease processes we're working on to keep us up to speed on what's going on in that disease process, where the liabilities are, where the potential opportunities are that really inform how we use the platform. So the platform is a collection of about 700 plus cell lines from individual patients, some of which were developed at UCLA, some of which were acquired through the libraries that you can use to access these cell lines. And we have sort of a rule that we have 20 or more cell lines in a given panel that represents 19 different histologies. So the major cancers that we deal with, all with the exception of CNS malignancies and prostate cancer. Not because they're unimportant, but because we don't have good models for them that fit into this platform strategy. But everything else we do have good models for. All these cell lines are characterized for their ability to grow in vitro, in vivo, as xenografts, and they're molecularly characterized for full genome sequencing, transcript array analysis, proteomics, to tell us what the expression patterns are, what mutations are, what gene loss and gene gains are, all in these cell line panels. We pair this data with public data from the things like the TCGA and other data sets, and we use a very simple algorithm that informs us about how to identify targets, and it's all shown in this panel on the right. If you start with all the proteins that are expressed in human cells, both normal and malignant cells, on the surface of cells, there are about 4,700 annotated proteins to the surface of human cells. If you now apply a filter and say, I want something that's expressed at high levels in malignant cells, but at low levels or not at all in normal cells from that same tissue, you go from 4,700 down to 800. Now, if you apply a more stringent filter and say, I want things that are expressed at a low level or not at all in any normal tissues, or if it is expressed in a normal tissue, it won't be a critical tissue that would lead to significant problems if we impact it. You go from 800 down to 200. And then if you say, where do the models we have fit and really mimic what we're seeing in the clinical databases from things like TCGA, you go down to 50. You apply the final filter on the bottom, where is there unmet medical needs? Where can we make an impact where our current therapy is not giving us anywhere near what we need for this particular disease? And you go down to 20. Those are the only biases we apply when we identify and validate the targets. And what came out of this program at the beginning were a series of 4 targets that look very encouraging as potential targets. And the very first and the lead one is a target called Claudin-6. It is a tight junction protein that is, for reasons that we don't quite understand completely, in some tumors is overexpressed at massive levels specific in the tumor cells. What you're seeing here is TCGA data. It's hard to read along the bottom, but I'll just tell you briefly. These are RNA transcript arrays. Each little circle is an individual tumor. There are collections of as many as 1,000 or 1,400 tumors in some of these different histologies. And the higher you are on the scale, the more expression you have of that gene in that tumor. And about a third of the way in sort of pink, you see the breast cancer group. About the vast majority are all located near the bottom. And there are a few that are tailing up at the top, but not many, and certainly not significant enough to chase that as a target in that disease. But if you get out to the far right, you will see testicular cancer. Almost all those tumors express extraordinary levels of this, but we have good therapy for that in terms of combined chemotherapy can cure about 90% of those patients. 10% this could be effective for. But right next to it, second lane in from the right, is high-grade papillary serous ovarian cancer, for which we do not do well. 85% of the patients ultimately succumb to this disease despite our best available approved therapies that we have today. And half of those patients express extraordinarily high levels of this protein. Now, if we look at does it make any impact on outcome, in the next slide we saw something very striking. This is a Kaplan-Meier curve. I think many in the audience know what this means, but basically this is a survival panel of expression of the protein. In black are the patients who do not have this protein at high levels or not at all. And in a disease where overall the outcomes are bad, you can see patients over time succumb to the disease as the curve gets lower and lower, and how many are remaining is at the end. But in red are the ones that are positive. So even in a disease with bad outcome, this identifies a super aggressive subtype. This looked very reminiscent of the HER2 story that we used to develop Herceptin in collaboration with Genentech. And then when you go down, one more slide on the last one, Dave. If you go down to overall survival, you see the same phenomena, which is the bottom panel, the upper being progression-free survival. But the other filter is on the next slide that they put up, and this is the expression in normal tissues. Impossible to read along the bottom all the normal tissues, but suffice it to say that red line is below detectable limits for the protein. All the adult normal tissues lack this protein. So this looked like a very attractive target in terms of the therapeutic window. If we go after it, we're not going to be impacting any normal tissues, at least in theory. By comparison, when we launched HER2, we knew that we could get away with a naked antibody, which is what Herceptin is, because a naked antibody doesn't carry a payload. And Herceptin has been very effective for patients. It's been a successful drug that's treated over more than 3.5 million people. That science came out of UCLA that initially identified this as a unique subtype. But all adult tissues express some level of HER2. The subtype of breast cancer we identify express extraordinary levels, which gives the therapeutic window. It's 2 logs off this chart. So that's why we could do it with a naked antibody. But with an ADC, an antibody carrying a payload as a smart bomb, this becomes less attractive. So things like Claudin-6 shown in the upper profile is what we really like. And that's how we built the platform for other targets we're chasing. So this went into a large series of studies that we won't go into today, preclinical and animal studies that showed that this was selective for tumor positives. It did not hit tumor negative patients, it did not, or tissues, and did not hit normal tissues. And that led to the clinical trial that I'll turn over to Aran to explain.
Thanks, Dennis. So the program for the Claudin-6 targeting ADC of trials is called the Catalina program. And here you see an output, the waterfall plot from Catalina-1. And this has a mix of Claudin-6 negative patient volunteer tumors and Claudin-6 positive patient volunteer tumors. And below the x-axis is the shrinkage of the tumor when the drug, trial drug was given to the patient. So as you can see here, it's a very active drug. The individual horizontal or vertical bars are a patient each, and the colors are the different tumor histologies that we have introduced into the Catalina-1 phase 1 program. And as you can see, and Dennis makes this point, wherever we find the Claudin-6 protein abnormally expressed, it's an oncofetal target on a tumor histology, and we treat that with the ADC, we see activity in the clinic. Next slide, Dave. The blue on that chart was the ovarian cancer, and so our lead program with the Claudin-6 targeting ADC is platinum-resistant ovarian cancer. This is the schema for the design of our registrational phase 2 trial, Catalina-2. It's a global trial with about 90 centers, clinical trial centers all over the world in 3 regions. And on the left, you see the trial design. We're taking 3 doses into the first stage, which is the dose optimization stage. And then later this summer, we'll select 1 or 2 doses to take through to the pivotal or registrational stage for FDA purposes. On the right, you can see the patient recruitment curve. The green was the plan, the target, and you can see we're tracking ahead of schedule with the patient enrollment for this program. Next slide, Dave. And here's an example of another one of our Catalina studies. This is the Catalina 4 study that Dave mentioned earlier. This is trialing the IXO-V antibody-drug conjugate with other standard chemo regimens in frontline ovarian cancer. So success with this trial gives us the potential for serving patients as the first line of therapy in their disease if they're Claudin-6 positive ovarian cancer patients.
So I think what Aran pointed out is what sort of drove Dave and I initially in terms of the founding of the company. And the frustration that I had as an academic is I think many of us, many people in the room as academics have had the opportunity to go to industry or to take jobs in industry, but still wanted to stay in academic labs to be able to do different kinds of work. And the idea was, could we take these drugs up to a point where we would be involved in their development? And we were never able to do that. As Chancellor Frank said, it was all done things were done in discovery, and then it was out licensed to companies. And could there be a model that would allow us to be involved in the development going beyond just what the discovery happened in the laboratory? And that's what's been so gratifying. That was what was unique about the model that Amir and Dave were able to work out to bring TORL forward, where the university is directly involved in the discovery and the development process as part of the company. And on one of our first patients from the Catalina 4 trial is getting this up front. This patient was attracted to the trial based on the fact that she understood what the best available standard therapy was combination cytotoxic chemotherapy. And she had a very active life, has a very active life, and wanted to try to maintain that active life and was looking for an opportunity to have something that would allow for that. And she was our first patient in Catalina 4, which is this upfront trial, newly diagnosed, that'll tell us how to use the drug, not only in late-stage disease, as you saw in the waterfall plot that Aran showed, but right at the beginning where our chances for cure are highest. So we're taking that approach both in ovary cancer now, and should the data continue to develop in non-small cell lung cancer, we'll approach it in a similar fashion in that disease. So it's very exciting. This patient has agreed to go public with her story and talk about how it's made an impact for her. And her statement is sort of on the slide as it had expressed in terms of her willingness to join the trial and be part of this sort of experiment. I'll only end my part by saying these people are not subjects or experimental dots on a chart. They're colleagues in every sense of the word. They're very much a part of the research program, like all of us are, and our gratitude is to them for participating in the way they do, them and their families.
Switching gears a little bit from our lead program, which to one of our newest program that's in the clinic. So we have our newest program that's just going into manufacturing, but the newest program in the clinic is our GPCR hematology target. So Dennis is going to talk a little bit about the target and why we picked it and how difficult it was to drug and why.
So I'll be very brief. This is a multi-pass protein that passes through the cell membrane several times, 6 times, and you can see there are loops that are sitting outside the cell. That is what you'd be going after with an antibody-based therapeutic. It is a G protein coupled receptor. What's unique about these is there are a number of different drugs that are targeting these in terms of FDA-approved drugs, but of the 33% of the FDA-approved drugs, 33% of the FDA-approved drugs are targeting this class of receptors. However, it's been very challenging to do that with biologics. Most of that is small molecule inhibitors of their active domains and intracellular domains. But Ben Hofstrom in the lab, who makes our monoclonal antibodies, really did a wonderful job of screening a huge number of antibodies to find an antibody that would work against this receptor. When we looked at the expression pattern, similar to what we saw with the Claudin-6 and ovary cancer, non-small cell lung, and testis cancer, we saw this target showing up in lymphomas and acute myeloid leukemias. I think the next slide sort of shows that. You can see that in our cell line panel, representing all the different histologies by color. Again, I apologize, you can't read the bottom, but it's all those 19 histologies that I told you. They're all very low levels until you get out to the far right, and you see 3 that are popped up at extremely high levels of expression. Those are diffuse large B-cell lymphomas, myeloma, and acute myeloid leukemia. So that would be the target for this, and I'm excited to say that, again, and consistent with the TORL model, we've launched the clinical trial for this. Our first patient has entered in the phase 1 trial for this and has been treated, and we're screening our second patient for that phase 1 trial. Aran can talk about the design of what we're doing.
And this is the design of that trial. So on the left, you see the staircase, that's dose escalation, and then to the right, the dose expansion to 4 different dose expansion cohorts. In the escalation, the volunteers are patients with relapsed and refractory non-Hodgkin's lymphoma, and then we have a variety of lymphoma and other histologies on the right. When you expand, we expand in the protein-positive target histologies. Obviously, because we have novel targets, we have to develop at the UCLA lab. We're developing not just the antibody-drug conjugate, but we also develop our own diagnostic. So we will be using both of those in the expansion, as Dennis mentioned. We've started this study, and we subsequently plan to build off work that the UCLA lab has already done, demonstrating that there is synergy, or appears to be synergy, between standard chemotherapies for lymphoma and this molecule that we're introducing into the clinic. We will plan to do combination regimens then as the next phase of development.
So again, switching gears to what's next. So we're going to just very briefly talk about our newest program that's starting manufacturing in the next month, but also imagining what could come out of the Slamon lab in the next year or two that could go into the manufacturing and could go into the clinic. Again, just reinforcing that the model that we've tried to build is that this is an engine that we keep turning the crank on, that we keep on looking for opportunities to make an impact in cancer. We keep investing in those opportunities, and some cases things are going to work, in some cases they aren't, but we're always looking to the next thing. So we're going to continue to invest in things that are working, but we're also going to continue to invest in things that we don't know whether they're going to work or not. And I think we heard a lot this morning from Lily's perspective on that. And even a small company like ours, and very small, we are looking ahead. We're trying to stay looking 5 years ahead and investing in 5 years from now. It takes a lot of discipline, and it takes a lot of investor support when you're not making money yet, which we're not, right? We're taking investor dollars and spending them on these programs. And so our first program is what we're calling TORL 5000. We're liking that. That's it. There's an inside joke there, but we added a 0 to the length of our normal programs, and this is 5000. But Dennis, I don't know if you want to talk just a little bit about this program.
Basically, this is a target that's come up that looks like it's expressed at extraordinarily high levels in gastric cancer, pancreatic cancer, and non-small cell lung cancer, a subset of non-small cell lung cancer. You see the percentages displayed on the slide here. This made it, again, an attractive target. In terms of the normal tissue expression, it's very favorable in the sense that it's negative or below the levels of detection at the majority of normal tissues and those that would be critically worried about. So this has gone into the antibody production now in the research-grade antibodies. It's been tested in the models that carry the target. It's been shown to be very effective. Tolerable in those models and in those preclinical animals. And this is the one that Dave's mentioned has now been elected by the company as the new IND that will go into production in a few weeks by our CRO that we use to make our antibodies for commercial grade antibodies for testing. An additional target is a program that hits an unmet medical need in terms of triple-negative breast cancer, which is something that we're particularly attracted to because we've had some impact in identifying the HER2-positive subgroup and then treating it with Herceptin. And then the CDK4/6 story has really successfully treated a number of patients with ER-positive disease, but triple-negative disease continued to elude us. Well, this target popped up as being positive in triple-negative breast cancer, as well as a separate subgroup of non-small cell lung cancer that you can see here, about 25 to 30%, as well as pancreatic cancer, again, a different group than our other target that's been identified in pancreas. And then we have a program in urothelial cancers where targets popped up at extremely high levels in about half of bladder cancers and about a third of squamous cell lung cancers, not the non-small cell group, but the squamous subgroup in that, which is, again, an unmet medical need, as well as some kidney cancers. So again, when Dave talked about a multi-asset company, when we see targets like this, we've been able to do the discovery work, we've been able to do the validation work, but without a company like TORL, we were stuck in terms of development. Now we're able to do that and still be involved as a UCLA program. And we have other programs that are going forward in terms of melanoma, where I think many people feel with the immune checkpoint therapies, we've conquered this disease. Well, nothing could be further from the truth. It's been remarkable that the checkpoint therapies have had the effect they had for about 20% of the patients. But the other 80% of the patients, once they progress, don't have any effective therapy for them. We have a target that now is expressed in more than half of the melanomas, including uveal melanomas, which is a huge unmet need, where we can chase that. So that's going as a potential target going forward. And we have a 10th program, again, a different subset of B-cell malignancies that could be accessed. So the work that goes on in discovery labs can now be actually actuated into a development program by this unique model that Dave and Amir were able to work out at the beginning, and that I'll finish my comments by repeating what Dave said at the outset, thanking the administration of the university with John, Jonice, Steve Dubinet, Roger Wakamoto, who really stood behind this company when there was a lot of naysayers and saying we should just take one target and go forward and not worry about a platform that's multi-asset. And so they stood by us. And then Jagdeep Bracker, who is at UCI, who said he also believed in the program and stood behind it. Without that, we wouldn't have had this possible, but it is possible for academic labs to do the kind of work we've been talking about. So I want to thank them for all the efforts they put forward. And of course, Dave and Amir for the initial development of a unique structure that happens between a private entity and a public university, which is still unique in the country.
You know, what we've tried to show today is that we are creating. I shouldn't say we've created, right? This is truly a work in progress. Our most advanced drug is in phase 2. We still have a phase 3 to look forward to and beyond that. And we have a lot of other drugs. But so far, we are creating a value creation engine for our different stakeholders. And we define those in this way that we've talked about. This is a version of a slide that we show to our investors regularly where we talk about our 2030 vision. And that's really to be commercial with at least one program, but to have multiple assets addressing histologies across the cancer unmet need landscape. It was hard to imagine 6 years ago that we would be where we are today. We're very proud to be where we are today. And in some ways, maybe it's hard to imagine being vertically integrated in just another 3 or 4 years, but we're investing in it, right? We are signing POs for $50 million worth of drug, which essentially will be commercial drug for XOV right now. We are looking to build a sales force. We are looking to do everything that you need to do financially to support all of that. But we're also thinking about the model and the optionality that we talked about. Not all of the programs are going to be successful. We've seen that already. A couple haven't. We did deprioritize. But not all of them will be programs that we take forward. And so having the option of exiting them to people who can take them forward makes a lot of sense. So again, we want to create drugs for patients. We want to do it at scale. and we want to keep doing it. And so really appreciate the time today in explaining the model. And hopefully what Chancellor Frank talked about this morning is something that this could be a model for how you can overlap what's done in the university and what's done in the private space. And this could be scalable in that way as well, in that it's scalable for other researchers, for other people at UCLA, in the UC system, and beyond. So thank you.
Thank you. Hello, hi, I'm Carla Zepeda. I lead the research and industry, the Industry and Research Alliance team here at TDG, and we just want to welcome you once again. My team is responsible for collaborating and securing research funding from industry. We've worked great with TORL, and we've had a great partnership with our next speaker. Again, we're excited that you're here. Please take the time to come see any of our team members that you see walking around through the conference. We'd like to thank you in person. Now, I'm happy to introduce our next speaker, Brian Bradbury, VP and Head of the Center for Observational Research at Amgen.
All right, good morning. So my name is Brian, and I work in the Center for Observational Research at Amgen. I'm also a faculty member here at UCLA, and I'm delighted to welcome everyone to this year's LABEST. So at Amgen, we've been privileged to have a long-standing and productive association with LABEST. We're excited about the work the organization has been doing since its inception in 2018 to build Southern California's reputation as a hub for biotech innovation. So LABEST gives us a very valuable opportunity to build relationships with emerging biotech talent across our region, to identify new partnership opportunities, to foster the next generation of scientists and researchers, and to help them translate their work into innovation that benefits patients. So Los Angeles has been Amgen's home since its founding in 1980. And over the last 45 plus years, while we've expanded into 100 countries and our patients now reach nearly 17 million patients annually, this is our home, this is where our roots are. And we're thrilled to be able to work with many here in the community, stakeholders like UCLA, USC, Caltech, and the Lundquist Institute, to help develop the best talent and invest in scientific innovation, all helping to support our region's vibrant biotech ecosystem. So we're excited at Amgen. We have recently invested in a near $600 million Center for Science and Innovation at our Thousand Oaks campus, which will bring together researchers, engineers, and scientists across multiple disciplines to enhance collaboration and accelerate the discovery of next-generation biotherapeutics for patients with the most serious diseases. Through the Amgen Biotech experience, we bring hands-on biotech experiments into high school biology classrooms, helping teachers ignite in their students a passion for science. Here, in Greater Los Angeles, the Amgen Foundation has invested more than $8 million in the experiment since 2002, bringing it to approximately 430,000 students across more than 330 schools. Additionally, the Amgen Scholars Program gives college, undergraduate, and graduate students opportunity to pursue summer research opportunities at top universities around the world. In LA, we have funded nearly 700 Amgen scholars, enabling them to take up research assignments at UCLA and Caltech, both of which will again host Amgen scholars this summer. As a final initiative I'd like to highlight for you is Amgen's SoCal Golden Ticket program. Since its launch in 2021, this competitive and selective program has given access to bench space, lab equipment, and scientific and business mentoring for 4 promising biotech startups in the LA area. The winner of this year's Golden Ticket will receive 1 year of free lab space at the BioLabs LA at Lundquist Institute, access to the Institute's other services and amenities, and mentorship by key Amgen scientists, and access to talent and expertise across Amgen's R&D network. If you would like to learn more about the Golden Ticket program, I suggest that you check out the LA Innovation Ecosystem panel discussion at 12:45 later today. So once again, thank you. Welcome to this year's LABEST meeting, and thank you for everything you're doing to help Southern California become a world-class hub for innovation in the life sciences area. Thank you.